and check out composition of the ash.
So should we try grabbing something here? By the way, you sound much better. I know. Just a reminder that if we do want to pick up a rock here, it will be our last rock for the dive. And we've got about an hour left. So what do we think, geologists?
I'd sure like to hear what Bob has to say, but I think all of this is sediment. What I'm a little worried about is if we do get to the break and slope, then the flatter area will be more heavily sedimented.
Yeah, were there 10-meter contours that are
That's my concern as well, Patty, that if we don't pick up a rock fairly soon, once we get up over onto a flat sediment, we're not going to be able to get anything.
Resolution, so they're kind of...
Right, so I would say, try for something.
I'm just wondering, they say we're going to go over a break in the
obviously,
relief.
these are not in situ.
Even if they're messy, could you turn them off for seconds?
Sure, let me take
Yeah,
a look.
with a piece of some of the
Because all we see is flatness.
impact
By the way, our ship
layers
should be
could
stopping.
be broken off.
Already.
If there's sediment, that might not be too hard.
Okay, I'll relay that.
Knots to,
Yeah.
you know.
Yeah, let's...
So pilots, do we still have time to try to take a rock sample?
Yeah, bridge moving the lead. Oh.
So in an ideal world, if we could break off a piece that's actually on one of those sort of layered sections, that would be great. But if not, we could totally pick up one of these guys that are just hanging around.
Or do you want me to go ahead and do it, pilot? Co-pilot.
Okay, so
That's a technical term, hanging around.
what looks good to you, actually?
On the very right, is it possible to, and now it's sort of rolling towards the center, is it possible to just grab a chunk of what looks like that's, sort of looks like it's intact? Is there any way to get an arm under that and grab it?
What?
Because we think it might be sediment.
You're talking about? What are you
Sort of
talking
right in the middle right here. We didn't see any, I don't think we saw any overhanging.
Definitely.
I mean, all these, I don't know.
Patty, do you have an idea of what you might like? Because I'm not seeing as many of the platey looking layers that we could grab.
That one to the right of the lasers is
This guy?
very bottom middle screen. It's a large
And we've got a
chunk
little
of something.
anemone hanging out to the right, just while we're waiting for this rock to be collected. Just note it, people.
So if we swing back left, do we see any of those layers anymore?
Not
Not really.
really.
Like in the top left right now? Not the big one, but the one below it. Actually, now it's dead center in the middle. Is it possible to crack off a piece of that?
Well, if we're able to crack it off, it's probably going to be soft, sediment-like.
We're okay with that if we could just get an intact sample. But if we keep crushing them...
Oh, you're okay with picking up a soft one?
Yeah, as long as we don't crush it.
Oh, okay, that's fine. I can pick them up gently, and not crush it. I'd just rather not break a piece off of the wall, if possible.
What about that piece just above that layered section, so on the left side of the screen right now? Do you think that one's loose?
Which piece?
The right below your
The lasers?
lasers right now. Do we think that's loose,
I
or do we
can
think
poke it.
that's part?
Ready for some mini Zeus?
Three full wide. Yeah, thank you.
Nope, we were partial.
Nope.
No, I was trying to get the little one above the massive one. Or crack off a piece of the massive one right there.
Like that guy?
Patty, would that be okay?
Yeah. It's got an open mic someplace.
UHECC, is Patty there? Can she confirm that this would be a good rock sample?
Abbreviations.
Hi, yeah, I think that's definitely, and if you can grab it, it's already pretty friable.
Yeah.
Got a light touch.
We'll give it a try.
I don't know, it may crumble when you pick it up.
Okay, well, we'll try to see if we can pick it up, and if so, we'll try to get it into a box.
Ready for a port wing inboard?
Yep.
Thank you.
Let's keep our fingers crossed. So they're opening the port forward rock box.
I can tilt up a little if you want.
So while the pilot's collecting this this rock sample, to try and ease the tension in the room. Oh. Okay, maybe no easing.
No. All right, Patty, contingency plan. Do you want to keep moving?
I know.
It's just so
You got
friable. I don't know that.
animal gallery online. Yeah.
Any of it.
All right, I think we're going to have to just move on from this. Are we ready to go?
Yeah,
Or
we're still waiting on the ship to move.
Okay,
so
well, if you're still waiting on the ship, then we can try picking up one more
I
thing.
have like a moment, and then I have to move, though.
Any time would be great, but if you have to go, you have to go.
Maybe there were a bunch, just to starboard and back, there were a bunch of like 10 centimeter cubes. They weren't part of the ground, but might be strong enough to pick up.
So again, easing stress.
Gonna try this piece.
Obviously, the Marianas is known for having the deepest spot on the planet. But not many people realize that there are 5 other trenches worldwide that are also deeper than 10,000 meters. No, not 5 others. 5 including the Marianas. Sorry, my apologies. So it's the Marianas, the Philippine Trench, the Tongan Trench, Kermadec Trench, and the Kuril-Kamchatka Trench, which is off of Russia.
And for those in the chat room, as soon as we're done with this operation, which might actually be successful, we will ask about time left on bottom and distance to the waypoint. Did it make it in there? It only broke in half. Excellent. So,
Well done.
Is this?
It looks like, yep, we just collected. Yeah, that's the port forward rock box, right? Yes.
Correct.
Excellent. All right, we've got our second rock sample.
Nice and delicately
Thank you so much. You
handled,
guys did a wonderful
pilots.
job.
Yeah. Soft touch.
Hopefully it'll make it up through the water column.
All set.
Giving me all the hard samples today.
All right.
All set.
All righty. Well, from the geology perspective, we've picked up 2 rocks today.
All clear?
Yeah. So I should be pretty close to done my move.
And the
You are-
solar looks okay.
Yeah, you are behind me, but if you move quickly now, we could
Yep.
probably do the next move.
I think we can call the next move.
Okay.
So we have about 45 minutes
now.
left in this dive. So we'd like to try and make some
Could I
headway
give a
to get
30
to
meter
that
ship
soft
move
sediment
at
area.
220 degrees at 0.2 knots?
30
possible.
meters, 220 at 0.2.
Good copy.
Another one of those Caulifaker sponges on the side. We don't need to go look at it, don't worry.
Initiated.
Yeah, at this point, watch lead, we're mostly waiting on Sirius.
Okay, great.
So if we can stop and look at things, it's just a matter of Sirius getting ahead of us that I would be concerned about.
And Nav, how much more distance do we have to go to waypoint 1?
260 meters.
And is that achievable in the next hour? Or
No.
45 minutes? No.
We have 48 minutes.
Right, so that means we're really going to try and push on as much as possible during the next 45 minutes, please.
So looking at the contours, we're at the same depth contour as waypoint 1 right now.
Yeah, I was
I mean-
going to talk to you, co-pilot, after everything died down.
Yeah.
The 10 meter wasn't loaded. It's in there, but it wasn't loaded when survey was started, so I can stop survey if we want to risk that.
So as we're moving up,
that's
this rock
okay.
looks a little
50 meter
bit
in
more
there.
continuous. There's still
That's what that is.
Yeah.
sort of talus and sediment between them, or rubble. But it looks like we're getting back into larger blocks.
Well, the 50 is good. It shows us that we're at the same depth, or almost the same depth as there. So.
We can still see some of that layering, especially in the bottom lower left right now.
So that means soon, if that's the same depth as that, we should come
See a
over the
little
top to the sediment, I guess. Theoretically.
Theoretically.
Okay, come on in, video.
So another stalked crinoid that we're looking at. This looks pretty similar to the one we saw before.
Is that in the background there?
Oh, what? The little bryozoan looking thing or coral thing?
Yeah,
Is that what you mean?
yeah.
Yeah, I'm going to guess maybe bryozoan.
Come in on
I could be wrong.
Guy in the aft.
And this also looks like it has some commensal amphipods
Can you repeat that, Pilot?
living on its stalk.
Stand by. Can we zoom on the guy in the back?
Ah, yes.
So scientists are sure, do we think that's a coral or bryozoan?
You're not on.
Scientists are sure, do you think that's a coral or bryozoan?
Get the guy in the back.
Okay. There it is.
All right. You can come out.
Thank you, pilots.
Oh man, and look at those nice layered rocks. This is what we had been hoping, I think, before we got our second sample to encounter, so we could just grab a piece and run with it. But we still managed to grab a piece.
Partial zoom. Right, coming out.
So we've really seen quite a bit of interesting geology on this dive.
Right next near our.
We've been seeing a lot of this layered geology. There are many
Yeah.
parts that we thought might be mass wasting from further up the slope.
Nav, bridge,
We've seen
move
these
complete.
strange mesh,
Copy that. Can we go ahead and
mesh
get another
fracture
move
patterns.
at 30 meters at 220 at
And we've
0.2
picked up and
knots?
crushed quite a few rocks so far. So we're having a pretty fantastic dive from the geology side.
I'm going to also say that this has been pretty good on the biology side.
a
Lots of
quick zoom
different
on these
things that not a huge abundance of life, but that's
Bridge now.
what we expected.
Go ahead.
Did you get that last ship request, move request?
No,
So
please
that
repeat.
looks like
Can
two
we get the exact
sponges.
same? 30 meters at 220 at 0.2 knots.
Understood.
two
30 at
sponges.
220 at 0.2.
Good copy.
But as I was saying, we weren't expecting to see a huge amount of life
boys,
because
yeah.
life here isn't easy. There's lots and lots of pressure, obviously. There's no light. There's very little
Nav,
food.
bridge, move initiated.
The only food which kind of gets down here is particulate organic matter, which by the time it gets 5 kilometers down isn't very much. And then you may get the odd tree or larger bit of food, but we haven't seen any of that here. And it looks, maybe because of the terrain, like it wouldn't be able to settle anyway. And then also, it's very low temperatures. I'm not sure what our temperature is here now.
About 1.5.
It's about 1.5 degrees Celsius, so which is really cold. And then also another factor which sometimes deep sea animals don't have to deal with shallower is that we're actually below or around the carbon compensation depth. Carbonate, sorry. What am I talking about?
Carbonate. Yeah.
Below the carbonate compensation depth. So that means that animals that have carbonates in their body skeletons or in their shells have a much more difficult time preserving that. So it means that we don't really see animals with carbonates in their body, and if we do, they're very, very thin. So it's just not an easy life down here, especially in this sort of middle range depth of 4,000 to 6,000 meters.
And actually, speaking of the calcium, the carbonate compensation depth, we get it wrong together. That was one of the ideas, or it's a question that we have with the sediment that we're finding here, is what kind of sediment it might be. Hopefully that piece that we collected will come up to the surface without too much of a problem, and we can find out.
Let's do a quick zoom here.
So another sponge here, and what looks like a tiny little jelly in the foreground as well. I think this is the second type of jelly we've seen today. It doesn't look, no, it's definitely not similar to the first one. that we've seen.
So tiny. Yeah, looks like it just landed.
So Nav, how much more time is on the bottom?
Right,
We have about 35 minutes before we need to get started getting ready.
So the last watch has a half hour.
Yeah,
Could be
I mean
worse.
total of 51 minutes. But
I didn't realize our watch went this late. I thought we had a 3 hour in the middle.
I think it is.
we done
a
at 3 or
3 hour.
3:30.
Oh, it's 2.5. 11:30, 12:30, 1:30, 2:30.
For those of you that have
3.5,
joined us on
yeah.
the previous two dives, it's really amazing to see the difference in, the descent and ascent and how much time we get to spend on the bottom. For those of us that work in the deep sea on a regular basis, it's probably not that surprising. But for people who are just joining us for the first time, those deeper depths, it really takes a long time to get down here. We have a shorter amount of time on the bottom, and then we have to say goodbye and go on back up
Snapped in?
with our precious rocks and biology samples.
That's a rock. All right, come on up.
We're just about to the end of this ship move as
Stick
well.
in here.
Sediment.
Strange.
But we still have about 60 meters with Sirius.
Oh, Sirius hasn't. Yeah, I haven't moved much.
Yeah. Like it's kind of at the beginning
Hey,
of it.
coming out.
Another trace from an Enstrop noose.
Maybe that's the close.
Oh, and there's something to the right. Horothurian, maybe, on top of the ledge.
Take a zoom video.
Yeah, just a quick zoom will be fine.
Yeah.
So yeah, this looks like the same purple one we'd seen previously.
Nav, bridge, move complete.
Thanks, Bridge.
While we've got a little bit of time right here, I will just remind our shoreside scientists that once we come off the bottom, we'll probably have about a 10-minute break, and then we will commence with dive planning for the next day. So for those of you who are still awake, please consider staying on the line.
Now, co-pilot
And
watch
our
leads.
dive
Back with you.
for the next
Pilot?
day is at Enigma Seamount. It should be really interesting, and we're going to ask Patty to
So-
probably talk about it once
We
we're
took
on
a
our
s-
dive call.
We took a sample, and then so basically Sirius stopped, and then
Okay.
we have done a 60-meter move, which is really just starting.
Okay. 60 meters.
Yeah, but
Okay.
the ship has already stopped from that. So there's like what's the delay? Like a 15 or 20-minute delay now?
It's about 16 minutes total
Yeah.
till Sirius stops
I don't
moving
know if
after you've
people
moved
at
the ship.
the
Yeah.
University of
So
Hawaii
we've
could
already- the
hear,
ship's
but
already
the
stopped, and Sirius
audio
is
was
just barely
very
moving.
broken up on the last bit of information.
It's taking about 8
Sure.
minutes for Sirius to start moving.
Okay. So we got some
Sorry
time.
to hear that, Bruce. So all I said was that we
have
will
to
be
chain
having
together,
our next
but
dive
it-
planning
the chain
call
together only about
about
this
15 minutes
maximum
after we
distance.
come off the bottom. And we will be talking about the dive to Enigma Seamount.
Is there anything in particular we're looking for?
No, we're done with geology, so I think they really want to get. They think, I'm not sure looking at
Okay, I
HIPAC,
can
but
get that,
during the
but,
science call they thought there'd be a
it is
transition
breaking up a
from
lot
this
now.
rock to like flat
I'll let
sediment.
our
So they really want to
our
get to
audio
that,
and video person know.
Hope there'll be different biology that you'll take a sample of, maybe.
Cool. Okay.
Watch me this video. I'm on it.
Thank you.
So I guess we'll let you get out ahead, and then we can call another move to tack on to the 60.
Is that a stock or a rope?
That's a new one.
That's something.
Fallen stock or line? Can't tell. You want to do a snap zoom video?
You got some to the right, sure.
Could we look at corallimorph to the right after this? There's a big corallimorpharian to the right.
All right. Zoom out. You sound like you're interested. Corallimorph.
Oh yeah. Oh yeah.
Is that your first? Is that your first
That
corallimorph
means an
there?
enemy type
Okay.
animal.
All right.
Although it's not, but they look similar.
Okay.
Maybe an anemone
Yeah.
initially looked like it was a corallimorph. I can't, not sure now.
It does have the tiny little baubles on the ends, though.
Okay, go ahead.
No, but I think you're right.
Those look
Oh,
like
wow.
tiny little bubbles.
What's the white thing?
Hold on.
That's
I'm going to go for a tighter zoom.
So this to me does look like a corallimorph, possibly Corallimorphus genus.
Whoa, extreme close-up.
Yeah, and so the corallimorphs are particularly, they're very easily identifiable because of the tiny little white baubles at the ends of each of their tentacles. Makes them really easy to spot.
Corallimorph. Okay, remember that one.
Those.
Great, thank you, pilot. We're ready to move on when you are.
Okay, let's go full wide.
Nav, how much time do we have left on bottom?
We have about 30 minutes.
Do we need to bump up the
Okay,
speed?
thanks.
Or I guess we won't even see the change until we got to come up at bottom already, huh?
Yeah, pretty much.
Okay. Was that our last move then?
I don't know. What? Wait, did we start another move yet, Neff?
Oh, we
No,
haven't.
not yet. I haven't
yeah.
called anything in.
Like,
Yeah, I
I'm
think
just
we
going to let
could-
you get ahead. You want me to go ahead?
I think we should put it in because
Yeah,
Okay.
he's ahead now. I'm definitely moving. I probably have another 30 or 40 to
Bridge
go.
nav.
yeah.
Go ahead.
We go ahead and put another 30-meter move in at 220 degrees at 0.2 knots.
30 bearing 220 at 0.2 knots.
Good copy. Thank you.
So in terms of the biological sample, is there anything we're looking for specifically?
We just wanted to try and get as to the sediment area. I don't know if that's going to happen, but
Okay.
no, there's nothing particularly sought after. But if I see anything interesting, I'll let you know.
Okay.
Can we just, that looks like another benthic ctenophore. Can we just snap zoom on that stalked animal right in
Sure.
the front of us, please?
Let's do a snap zoom video.
Yep, another benthic ctenophore. Probably on a spun stalk. Oh, are those eggs? Maybe. Okay, that's great. We're happy to carry on as we
Okay,
saw one of
enough.
these earlier. Thank you.
Tara?
Watch lead. This is co-pilot.
Sorry, co-pilot, what's up?
I just had a question. I wasn't at the science call yesterday, but if you look at HIPAC, we're about at the depth contour of waypoint 1. So I'm wondering, was there a transition in backscatter that makes us think it'll be different, or?
So unfortunately, we don't have HIPAC up in the back anymore.
Okay.
But from the...
But
Okay,
if,
wait,
if
hold
you
on,
did,
hold
you
on,
would
hold on.
see.
Geologist just cruising in the room to answer that question. Bear with us. So this is another Amperima peniagone type holothurian that we saw earlier. It had the quite large feeding appendages and podia.
It's that, oh, I think it's one of those swimming worms, upper right.
Yeah, do you want to do a snap zoom video when I frame it up?
Sure.
Yeah, looks like it.
Oh, that's cool. Good shot.
Navigator, could you move high pack a little to the left so we could see a little bit further?
Sure.
So Scott France was saying that it's interesting that we've seen another one of these benthic ctenophores with its sort of fishing lines extended. And that's implying that there's obviously something in the water column that they're catching, enough for there to be two of them in this small area.
Sure.
Scientists at home, is there another biology sample that's kind of caught your eye along the way? Is there anything in particular you'd like us to look out for?
I think if we saw another one of the sponges, either the Caulophacus or that elongate one, that might be worth collection. The elongate one in particular has need to confirm that ID. I'm pretty sure it's a demosponge.
Yeah, the elongate one is a lot more enigmatic, I guess.
If we don't happen across any, it would be a good idea to collect one of the holothurians.
Any particular holothurian, Jeff, that caught your eye? Jeff? Okay, well, we'll keep an eye out for holothurians and sponges. We are in the last 25 minutes of our time on the bottom. Sorry.
There's a bit
Jeff.
of a delay in the audio.
That's okay.
I think that if you don't... So if you have a chance, go ahead and get a cucumber. You're going to holothurian, if not a sponge.
Are the sponges we're looking for stocked, or?
So I guess the next sponge, all the ones we've seen so far have been stalked.
Okay.
So either a stalked sponge or a holothurian.
Okay.
I think they prefer the stalked sponges, and there were two types we've seen on this dive. One looks kind of like a...
coming up. Up ahead.
Yeah, that might be the kind we're less interested in.
Okay.
There looks like there's one down.
We can
right on
cruise by it.
Yes, go about center screen towards the right side. It's kind of small. I can do a partial.
Go ahead.
But I think we'd like our next sample to be a sponge, please.
Okay.
Top of the screen.
Co-pilot nav.
Oh, unless that's a jellyfish. Okay, never mind. It's a jellyfish.
Go ahead, Nat.
How long have you guys been pausing in between each move? I don't see any cliffs or anything.
Saw another stock up on the right.
Andy's been doing a good job. We've pretty much only paused when they're looking for a sample.
So just keep them kind of going and.
Kinda, but there's been enough zooms and samples, and we've gotten behind enough that we
So
have
there's
been
a
pausing.
couple of sponges coming up, but also Casey pointed out that there was a fair amount of interest from the guys in Guam for holothuroidian.
Okay, thanks.
Which just showed in view.
Yeah, thanks.
Right, so there's a holothurian and a sponge I can see right now. Of those two things, which is of higher priority?
Yeah, that's the way to waypoint one.
We're going to end up collecting, I think.
You think you're going to end up collecting one of these?
Yeah.
Oh, okay.
Talking about it right now.
All right. Then I'll let the ship settle.
Yeah, I still might have...
You've got 8 minutes or something or 7 minutes of swing left.
I think purple wins out over white any day.
Okay. We're going for the holothurian?
Okay, yeah, so if we could try and collect this holothurian, I don't know how easy or difficult that is. No pressure.
We'll find out.
I will say, if we think that we're going to damage this beyond repair and not be able to pick it up, let's not do that.
I think we'll be able to get it.
It certainly depends on whether it's muddy underneath. If he can scoop it up rather than grab it.
Okay.
But he might start swimming, I suppose. If he starts swimming, don't chase after the hand. That's going to be...
Yeah.
If you just start flinching, like grabbing out in the air, that's more likely to cause damage. If he stays nice and
It is
still
very
for it,
exciting.
I mean, he looks kind of manky, though.
Really?
Can we... Oh, before we collect him, we need to get excellent imagery, please.
Sure. I'll just set down and we'll zoom in. So we get the hydraulics ready.
Poor guy doesn't know what's going to happen to him.
No. It's like that crinoid yesterday that
Pilot,
just
just let
hitched
you know.
onto the ROV.
We have a live
And
crinoid in the
then
left.
mid-ascent,
port
probably
side
just like
there.
dived
Okay,
off
thanks.
as it got too
So-
hot.
So I was on an ROV cruise once that had a
live
mesh
crinoid.
bag
Live
across the
crinoid.
front,
So we're gonna go
and
for
one of
the
those
starboard
big giant
firebox.
crabs
Yes.
hooked onto it and would not let go. It at the Wanda Fuca.
I guess I gotta
And
move
that
it
crab,
over here.
the big orangey white ones, yeah. But and it like grabbed onto the outside and it came all the way up with us. We were like, what are we going to do with this crab? It's not what we study.
And then you preserved it.
I believe we did. Actually, I think it went into the freezer, if I'm not mistaken. I think
No
someone just
way.
put it into the freezer. We didn't have anyone who was studying it on that
Do you want to
cruise.
zoom in, Tara?
Wow.
So, just something to note, make it a little bit more difficult. Holothurians can swim, some of them.
My
So,
navigator was just telling me.
yeah,
And can't some of them spit out their body parts
they can.
internally?
I mean, I don't know about deep sea ones. Shallow water ones definitely do eviscerate themselves, basically, as a...
He won't be a very fast swimmer, but he will
So this could be a very
He does do
exciting
that.
collection.
wave in the water.
Swimming,
off.
guts flying.
So should we,
That's a rocky bottom.
yep,
I don't know if you're going to be able to scoop.
might be a little more sediment on the
He's a good
the
size,
inside of
too.
this, so
I'm
if I
excited
angle the
to
hand downward and from the front.
That's true. What's it?
play with that one.
25 centimeters long?
If we do get it.
Okay. Let's prepare the starboard bio box.
Just watching for Sirius to settle.
Sure.
Yeah, she's coming forward a little bit yet.
Co-pilot, do you know how to set up the starboard bio box for sampling?
Yes.
Okay.
Stand by. Just gotta winch
Yeah.
up a little bit here.
All right. So 48.
Yes. Bio.
Spooling up hydraulics. 1600 RPM, good enough?
Yeah, that's fine.
Two hours, 40 minutes up. We need to be up at. We're at.
So it'll just be another minute here. Watch lead. We're just getting set up. Would you mind handing me the binder over there?
Say again?
The predator binder. I'm gonna-
So one of our echinoderm
Yeah,
taxonomists
there's
on
no
shore is saying that a fused brim, like what we're seeing on this holothurian, which is the kind of collar around it, that means that it's a swimming holothurian.
Okay. How are we looking, co-pilot?
This is really when you just need one of those like butterfly nets.
The drawer is ready
Or
to
slurp.
extend.
Yeah, or a slurp
Okay.
gun.
Just pulling up a little
Just
bit
catch
here.
them.
And that was
the
Alex
winch.
Kerrigan
Okay.
from
So Sirius
University
is
of Guam
still
Marine
swinging
Lab.
a little bit.
Thank
so
you,
we'll keep
Alex.
an eye in.
Okay. You want to wait or?
No, we'll
Okay.
we'll just keep an eye out. That was just one 30-meter move, so
Oh, okay. Okay.
wasn't two in a row or anything. So shouldn't.
All right, co-pilot, are we ready for hydraulics?
Yes, hydraulics are on, spooled up.
Hydraulics come on. Go grip force 1. Okay, arm coming on.
Are
can we
moving
get
a
the
little bit,
you want to
sliding
wait?
forward.
Oh, yeah.
See if it settles. There you go. Alright, just locking auto heading. Just in
So we've
case.
had a tentative ID for this Holothurian, and I agree with it, flicking through the OER Atlas. Chris Ma said that he thinks this is Galatheothuria, which we also saw before, and ID'd to that as well. And that's in the family Synallactidae. And hopefully we can get him on board and get scientists to potentially confirm that ID in the future.
Okay, let's try this. Zeus isn't going to help much. Hey, co-pilot.
Go ahead, co-pilot.
No
Get
pressure
the
on our ROV
starboard
pilots right now.
Manipulator camera up on the main screen, please. My screen. Oops.
It's on the lower row. There you go.
That's starboard minute.
Yes. Thank you.
So no pressure, ROV pilot, but if we do get it, the guys at the University of Guam say that this will be a fantastic specimen to add to collections and ongoing research there.
Don't tell them that now. Tell them that after they've collected.
No, I want to put the pressure on.
So you shouldn't have said no pressure.
This family, that is true. I should have just said lots of pressure, guys. So this is from the family Synallactidae, which is polyphyletic, and so it'll be interesting to do some molecular analyses on this to see where it
touch them.
Sort of ends up.
Right before you go.
So,
I see.
Yeah, exactly.
It amazes me that this Holothurian hasn't tried to escape yet.
Yeah, usually you sit down and they take off.
Yeah, if it was me, I'd escape.
Don't clamp too hard.
Okay.
Don't look. So folks at home, I mean, this is what we do. We try to pick up things off the seafloor. Sometimes we're successful, sometimes we're not. Hopefully this will be one of our more successful biology collections.
Thanks for the qualifier there.
And once we get it back onto deck and to the repositories where it's going to live, scientists can use this. Scientists from anywhere can access this specimen. It will be housed at, I think, the Smithsonian.
Take a look at your mini Zeus
And,
real quick, where your arm is.
and scientists anywhere in the world can access the specimen and use it for their
I was hoping
analyses.
to go more from the front, but I think coming from more from the top is going to work better. Because there's in a little bit of a dip there. Let's get that box open. Shoot.
So this is just
We got a
showing
hand
the
on it.
skill of our pilots here on board.
Oh, what are those body parts? Oh,
Oh,
is that
God.
eviscerating itself?
Or it's poop.
Oh, it's poop. This is
No, that's definitely poop.
well. Now we know what Holothurian poop looks like.
It's basically just sediment. So they deposit feed, so they'll eat lots of sediment and then take the nutrients out of it, basically, and then just poop out the rest of the sediment.
Folks at home, you've now just seen your first holothurian
pooping on camera.
Oh, we were just talking about what it's doing.
Okay. I mean, if something came down to the deep sea and grabbed you,
you'd probably poop.
You'd probably poop too. Exactly.
Would you like me to
so
adjust
outboard?
Yes, please.
Okay.
If you could zoom in, Tara, that would help.
It's disgusting.
Stand by.
I think.
All right, so we have a final step. The most important and critical step is getting that sample into
I am
the box.
so excited for the sample.
Okay, so just so you know,
Not responding.
Yep. Just a thought. He might try to swim.
So we got to close. I'll get the arm clear, and
so if you actually fly up with the ROV and force some water down in the box as you let go, then it might.
These guys think of everything, you know.
The drawer, not be less likely to float away.
It's because they're experienced pilots who have done things like this before.
Serious, that might be something you can do. So he's definitely kicking.
Oh.
I think I prefer maybe just to. You think he's going to swim away?
He's swimming right now. You can see his head going. So if you can try to let go and see what happens.
No, I mean.
If, okay, hold on. I can do this. Oh. All right. So I'm going to come out of joy lock and float up.
If you just come up, yeah, and you can either put it in auto depth and set it a little higher than you are, so it just goes up right as you let go, and
I'll try to
just push
close
up
the drawer as soon as you're clear.
You can just give a good push up and see how that.
I'm going to put it out of joy lock, give it a good push up.
Yep.
And go with that.
Yeah, I think the force of water down will keep them in the box while you close, hopefully.
Okay. Ready? Here we go.
Perfect. I'll go for it.
Close it up.
All right, let's see if you can close it.
Excellent job, guys.
Press the button.
So there you have it. We have a holothurian collected without a slurp gun.
That's why you put away.
Just a heads up.
Casey, it went into the starboard inboard box. Inboard box.
Can we get the main Zeus up on the camera?
Oh, they're all clapping.
So you're getting huge applause from the UHECC as well as many compliments coming in via the chat.
We'd call it a
ROV
standing
pilots.
ovation, but we don't know if they actually stood or not, guys.
How does that arm look, Jeff?
Yeah, I mean, try picking up on your shoulder a little more. It looks like it's dipped down. On the shoulder.
Yeah, I think shoulders.
Okay. It's just back toe.
Yeah. All right. Hydraulics off.
Okay, so we have about 10 minutes left in our dive. So if we have any more time, if we could have a little drive around before we leave the seafloor.
Sure.
Great work again. I am so excited to play that holothurian later.
Jeff, is it best
all
to
25
disable
centimeters
HV?
of its purple deliciousness.
Yeah. Okay. Can I switch your right camera, pilot?
Yeah, go for it. Thank you.
Yeah, I'm looking all the way down.
All right, I'll push out ahead.
Copy.
Thanks for your assistance, both of you.
All right, let me know when you start getting pushed out. I can call in another move for you.
All right.
Or actually get 8 minutes. So.
And we're moving up a fairly heavily sedimented slope. You can see small rubble kind of wandering down the slope. There's definitely still some intact rock.
I'll push out. Come port.
got a fault on your aft flap, just so you know.
Okay, thanks.
Sorry. Yep. Perfect.
Navigator, what's our status on moving? Did we just settle out? Now you're going to call one in?
Yeah, I'm about to call one in right now.
Okay.
Oh, can we see that coral to the right? This could be real important.
Coral to the right?
And I don't think it's a coral, I think it's a sponge.
Do you mean the... Yeah, so I think they mean that one up on the ledge.
Oh, okay. Oh, the delay.
It's behind you to the right.
Oh,
There's
okay.
behind us.
But while we're looking for that sponge, I have to tell you, this is some fantastic and incredible geology.
Okay. Well, the ship is stopped, right?
It was down below the ROV on the right side, lower right side. You passed over it.
We'll stay well above and back up. Tilt down.
I think it was to your starboard.
Yeah, keep spinning right a little bit.
Can you follow me down?
Oh, right there. That one down there.
Right?
Oh, yeah, okay.
There it is.
So this is a...
I think this is one of these really weird sponges that Mbari described. It's a cladorizid.
Yeah, I know exactly what you mean.
Sponge. It's really unusual.
Okay, we got lasers on it. It's going, Tara.
Mm-hmm.
And let's kill the lasers, co-pilot. We got it already.
So this is a cladorized sponge, I think. Oh, it's got lots of ophiuroids on it, living on it. Wow, this is so interesting. And the one that Chris was referring to, that the Mbari folks discovered in 2012, actually had 4 of these projections, so it was kind of like a cross, whereas this one seems to only have 2, but I think we are right in still thinking it is a cladorizid. And is 100% a new record for this area. And hey, could very well be a new species of sponge. Who knows?
Note in the event log, there's a clamor to collect if there's any chance of doing that. I know we don't want to open up that box again, though.
I don't think we have enough time. Yeah. Plus, I think we have to come off bottom very quickly.
Did the
crinoid?
We can get some great imagery of it if that's any consolation.
Said that was a concern, but
okay.
Zoom in, Tara?
Yeah.
And just a reminder, these sponges are carnivorous.
Sure.
Yeah.
They're carnivorous?
Yes, these cladorizers are carnivorous. And there are about 5 ophiuroids living on it commensally, or 6.
Are they dinner?
No. I'm not really sure how that's not happening, but they are not dinner.
Yep. Nice.
Nav, can I ask a question?
There we go.
Go ahead, watch lead 2.
So we don't have enough time to collect another thing, do we?
I'm doing the calculations right now.
And do we have a place
If we leave the bottom
to put
now,
it?
we'll get back at 1804. So we need to be back at 1830. So we have maybe 15 minutes till we want to go.
Do we have a place in the port bio box?
Are we afraid of that crinoid
No,
swimming away?
No,
I don't think the crinoid swim.
Oh, I don't know.
Some do, but it didn't seem like that one was
That one,
kicking
yeah,
when we
he
picked it
wasn't
up.
very. He was not very active.
So
I think it's better to open the crinoid one than the holothurian one.
Definitely.
But I think
Not
if we
'cause
do
you're
have
biased.
time, we should get this.
Let me just get the whole argument.
It would be the last thing we do.
Yes,
And,
which
yeah,
a lot of
we'll
the
have to make
scientists in the
quick work
chat
of it.
seem to be
Do it quick.
happy that it is the last one we do.
Okay.
Not Tara?
So you have 2 bio samples already? Is that
We do. So we'd like this. This needs to go in the port bio box
Okay.
where there
Yeah,
is
yeah.
one free.
And how much
The
of it are we
outboard.
hoping to take, Watchley?
So we would want to take the whole thing. Can you put on the lasers?
Sure can.
Please?
Put them back on.
The whole thing.
That's going to be tough.
Yeah, ideally we'd want it snipped from the base. But I think that's
Is this a brittle?
Are these brittle or not?
I have no idea.
We don't know.
We don't know.
Yeah, grab the whole thing if you can fit it in a box.
I'll tell you in a second.
What do you think, Jeff, if I come in from the top?
They're not as brittle as glass sponges.
You got
I'm
12
thinking.
minutes. What's that?
If I come in from the top, because I'm not going to be able to get the clippers in at the base.
You can try to get your fingers underneath the two arms if that's what you're thinking. Is that what you're saying?
Yep.
And then pick straight up.
Yep.
If that will unroot it, but it might break it in half and not unroot it. So the other thing you can try to do is, Josh suggested this yesterday when I had a failed grab, is just take a finger and poke near the stem where it meets the ground and sort of knock it over and then pick it up off the ground. It's sedimenty right there, so you'll be able to scoop underneath and pick it up.
And so from the chat room,
So those
I
are
believe that
those are your two.
if
I
we
don't
can
think
pick it
I
up,
agree.
we
I
will
don't
also
think you'll
take
be
the
able to get your
opioids
claws
with
under
it.
there.
So,
And if you do, you're
by
gonna
some chance it
crush
breaks,
half
we'll
of
try
it
to
with
take a
your
piece that has those
jaws
dimensionals.
anyway. So
And the scientist is saying that it shouldn't be brittle. Should not be brittle.
So maybe I should try to knock it over.
You can try knocking it over. And then, with that sediment there, you'll be able to sort of get your fingers underneath and pick straight up.
Pilots, is there a way to only take half of it? There's some concern about taking the whole thing simply because it's the only one we've seen.
Yeah, let's decide what we want to take and do it quick because we're running low on time.
Okay, Diva, it's up to you.
I will attempt whatever you need me to attempt.
I mean, one person saying take half, the other person saying we need more than one ranch. They're only, yeah. Okay, yeah, then maybe just try and rip half of it away, please.
Yeah, so in that case, yeah, I wouldn't even rip. I would come from the top, try to get your fingers in such a way that you can cut right where it bends there.
Whichever
Take that
half is more easy for
Take
you
the
guys.
Right. Oh, the left. Maybe the left, yeah. Okay. That's probably... Hopefully there's not too much rock underneath. Yeah.
Yeah, you want the starboard bio box?
The port.
Or port?
Yeah, by right we mean port.
Yeah.
Want the port bio box.
Yes. Got it.
So, do me a quick favor. Close the drawer all the way.
I just did this, but yeah, I'll do it again.
Okay, good. And then retract both pins.
Yep.
Fully. Give it a good long hold. Yep. And then only extend the port. Perfect.
So while the pilots are doing this, I can say that these carnivorous sponges, cladorhizids,
5 minutes.
they, as I said, are predatory.
Yeah.
So
if you want 10
usually,
minutes to set up, then you've got.
usually sponges feed by straining water through their bodies, and they catch bits of organic material and bacteria. But in this case, with these particular types of sponges, cladorhizids, which we also saw a different type earlier in the dive, it looked a little bit like a pom-pom kind of at the beginning on a stalk.
Yeah.
What these cladorhizids do is that they snare tiny crustaceans with barbed hooks that cover the sponge all along the limbs of the sponge. And once the sponge has its prey caught, then it basically envelopes the animal in a thin membrane and begins to slowly digest it.
ROV
So
HD1.
Accessing the software.
It is an extremely
Not HD2, HD1.
exciting collection if we do get a piece of this, and it probably will be a new species.
And then just focus it up and I'll be ready to go. Thank you. Alright, hydraulics coming on. Let's get the binder again.
Oh, yeah. Okay, you've got 4 minutes.
Okay. Alright. Hydraulics coming on. Grip force 1.
Ready to open drawer on your mark.
Thank you, co-pilot.